Hi, welcome to our showcase for grünif.ai. grünif.ai is our attempt to provide an interactive, AI-powered in silico compound optimization platform based on the continuous vector representation of a molecular structure introduced in our recently published paper in the Journal of Chemical Science. The optimization of a given starting molecule happens through a particle swarm optimization procedure, in which the molecules are optimized against a user-defined objective function, which can be an aggregate of selected molecular scoring functions, for example, ADMET properties or biological activity. For more detailed information on the utilized optimization method, please see our recently published preprint. In grünif.ai, the user can sketch or paste a compound structure of interest into the sketch editor, Marvin. Depending on the optimization problem and the desired compound properties, the user can select from a range of pre-configured scoring functions. These scoring functions can be extended programmatically by writing new ones that adhere to our standardized Python API. They can be arbitrary Python functions, or for instance, problem-specific machine learning models that take molecules as input and output a score. We will demonstrate grünif.ai using an EGFR inhibiting compound that we arbitrarily chose from Kemble. In this particular case, let's select solubility as the first optimization criterion and utilize a QSAR model that was trained on predicting the solubility for a given compound. By clicking on the settings icon, the desirability curve editor opens. For each scoring function, a reasonable desirability curve is pre-configured. This desirability curve defines ranges in which the predicted scores are favorable, approaching a desirability value of 1, or disfavorable, approaching a desirability value of 0. New points can be added to the curve by double-clicking the canvas, while double-clicking an existing point results in the points deletion. All points can be moved so the user can shape the curve as desired. Let's additionally add two structural constraints. A substructure match constraint can, for example, help to focus the optimization surrounding a desired scaffold structure of interest. By clicking on the settings icon, a molecule sketch editor dialog box opens, and a substructure can be pasted or sketched into the editor. During the optimization, generated compounds that do not contain this substructure get penalized. Furthermore, we can add a distance scoring function, which calculates and scores the distance of the generated compound structures from the input structure. In this case, I'd like to obtain structures which are somewhat distant to the input query structure, but still have some level of similarity. The sliders on each scoring function enable the user to adjust the weight or priority assigned to each individual scoring function within the global objective function that is being optimized. We are now ready to begin by clicking Start Optimization. The main calculation happens on a GPU cluster and can be parallelized over multiple GPUs. In our default settings, one optimization run consists of 5 update steps and takes about 30 seconds. All parameters can be adjusted according to the user's specific needs, for example, in the case of a different hardware setup. Once the first optimization run is finished, the results are visualized. The chart on the top presents the D-score, the weighted score over individual scoring functions on the y-axis, against the optimization runs, so that the optimization progress can be monitored. The generated compound structures are presented in a grid format. The colored circle on the left displays the compound D-score. By clicking on a compound card, the individual contributions to the D-score can be inspected. The left column shows the name of the scoring function, the middle column the predicted value, and the right column the derived desirability score as defined in the desirability curve for each scoring function. The molecules can be sorted according to the D-score, or alternatively according to the compound structure similarities using a back-end structural clustering algorithm. If a user is interested in a molecule, he can add the molecule to a basket in which all molecules of interest are collected. The user can also change the weights assigned to the individual scoring functions at any point to influence the direction of optimization. Furthermore, we have implemented a user preference scoring function to further steer the optimization. If a user wants to explore the chemical space surrounding molecules he particularly likes, he can click on the heart icon of the molecule and push the algorithm to explore compounds that are nearby in the chemical space. Moreover, by clicking on the ban icon, the user can push the algorithm to avoid the chemical space surrounding any undesirable molecules. By clicking on the Continue Optimization button, the next optimization run starts and all user adjustments are considered. The user can continue the optimization until he finds a sufficient number of molecules he would like to pursue further. At any time, or when he is finished, the user can export the basket of molecules and use the generated molecules in other applications, for example for docking or for synthesis planning. With this, we conclude the showcase of grünif.ai. We hope we were able to illustrate its functionality.